Welcome back to the class. So we were learning about various microorganisms that are used in human welfare. We spoke about various organisms that are used in our household products. It might be curds, idli dosa batter, or the bread dough. And then we were talking about growing these organisms in large fermenters and making beverages out of this, and also extracting this antibiotics. Here, I would like to add one thing, like saying that it's not just the antibiotics or the beverages for which we make use of fermenters. We make use of fermenters to produce various other chemicals, various other enzymes, and various other bioactive molecules, and all these are again produced in large, huge fermenters used for various industrial production, more importantly, the commercial production of certain chemicals. So in this particular class, we would be talking about some of the other most important products which are of commercial use and industrial use obtained from microorganisms. Along with that, we would also focus our study on some of the microorganisms that are used for uh, water treatment. So over to the first one, which is the use of microorganisms for production of various chemicals, enzymes, active molecules, so on and so forth. The first important component that we see in the production of with the use of microorganisms is the production of acids. You might be wondering why acids are basically required. When we talk about acids, we have two types of acids, the mineral acids and the organic acids. Mineral acids, I'm sure you would have studied in your chemistry that they are generally prepared in the lab, where you take group 1 with group 7 elements. You have sodium hydrochloric acid, then you have sulfuric acid, so on and so forth. All these are acids. Then we have organic acids, which are considered to be more natural. So formic acid, citric acid, then we have tartaric acid. All these are considered to be acids. And I'm sure you know why. We will not talk about the chemistry part, but we know why they are called as acids. Their pH and their ability to donate a hydrogen, all this makes them extremely useful for various purposes. Now, when we're talking about organic acids play a very important role, industrially. For example, I'm sure when we make lemon rice, we use citric acid, but not all the time we get that citric acid from lemon. Right now, we have this citric acid salt, and this salt is mixed in water, and we use this water to make lemon rice. In fact, we use the citric acid even to make certain juices. Now, where do we get this citric acid from? Now, this citric acid comes from some of the sources like fungi, which happens to be a microorganism. Have you heard of this organism called as Aspergillus niger? Aspergillus niger. Now, this is a fungus, and we see that this Aspergillus niger produces good amount of citric acid. Uh, In fact, many of the places we grow this Aspergillus niger in a conical flask, huge conical flask, of course. It grows and forms an upper layer, which we call as mat. Now, below that, we will have the liquid, the nutritive media, which will have high amounts of citric acid. So you can extract the citric acid, purify that, and use it for whatever thing that we want. So Aspergillus niger is one such nice organism used to produce citric acid. There is one more organism, I'm sure you would have heard of it, which is called as Acetobacter aceti. Heard of it? Acetobacter aceti, a beautiful small rod structured bacterium. Now this produces acetic acid. Now you might be thinking where acetic acid is used. We use acetic acid in making vinegar. So we use this organism in making vinegar. So this acid is very commonly used. Along with that, we have one more which is called as Clostridium butylicum. This is again a bacterium which produces butyric acid. And then I'm sure we all know this. The other one is the lactobacillus, which produces lactic acid. So you see that there are so many organisms which produces acid, and all these acids are being used for various purposes. Now, for example, citric acid has a lot of industrial applications, commercial applications. Acetic acid is used in food industry. Butyric acid is used in making certain drugs. Again, you also used in food industry, and lactobacillus again used in food industry. So acids basically are very important in food industries and has a lot of industrial and commercial applications. One more important product that we get from microorganisms is alcohol. Now, when I say alcohol, I would specifically refer to ethanol. Okay, ethanol is commercially produced in large fermenters uh, with the help of a microorganism which is called as Saccharomyces cerevisiae. Okay, Saccharomyces cerevisiae. Now, the Saccharomyces cerevisiae is that baker's yeast that we call. Now, this is being used for production of ethanol in a very, very large commercial production way. Also, I'm sure you have heard of the use of the certain enzymes in various products. If you have noticed your advertisements, they would say this particular surf powder has, I don't know if it is surf, just telling you, not promoting surf anyway. Uh, so this particular chemical, this particular powder has protease which takes out the proteins and the dag. They say all that. Now that is because along with the normal soap, they would have added certain enzymes. The third and the most important use of fermenters comes in the production of various types of enzymes. So we have various types of enzymes. The first one is a class of enzymes called as lipases. Lipases are those enzymes which dissolve and removes oil stains. So if your soap powder has lipases, even if there is oil present on the cloth, that gets digested because of the lipases, and the lipases action will make the clothes cleaner, right? So this is one very important enzyme. Along with that, we have a set of enzymes which come under the class of proteases and pectinases. Okay, proteases and pectinases. Now, what are these proteases and pectinases? So you know that proteases are the ones which takes out proteins. Pectinases are the ones which takes out pectins, which digests pectins. Now both of them are used in making our used in laundry, okay, to clean the clothes. However, these are also used as clarifying agents. You know what is clarifying agents? These are the agents which gives clarity to any solution or to any suspension. If you go to market and buy any fresh fruit juice, you see that it is turbid. You agree with me? Whereas if you take any packed food juice like Tropicana or Appy or whatever, and you look at the juice, you see that it is very, very clear, right? Now that clarity of that solution, of that suspension, comes because there is addition of some kind of clarifying agent. Both proteases and pectinases, both of them are used for making clarified juices. That is one more enzyme that is being widely used in various industries. Then there is one more enzyme which is called as streptokinase. You know what is kinase? Kinase is the one which adds a phosphate group. Okay. Now when it adds phosphate group, uh, you see the phosphorus in the solution comes down, and on this particular substrate phosphate is added. Now this particular class of enzymes, which is called as a streptokinase, is being used by medical uh, technologists. They have slightly modified this particular enzyme, which is the streptokinase, and now they are using it for those patients who who are undergoing treatment for heart attack. You know, whenever there is this myocardial infarction, there will be some small clots that would be formed, and these clots are digested by using the enzyme which is the streptokinase. Now you might be wondering, how does this work? Very simple. So there is a clot to which there is a phosphate group that is added, and this changes the morphology and it becomes liquid again. So it basically acts like a blood thinner and is targeted towards the clots. So it's called as a clot buster. So streptokinase, a modified version, a genetically engineered version of streptokinase, is being used as a clot buster. Just like that, there is one more chemical which is called as cyclosporin A. You know what is cyclosporin? Cyclosporin is a very, very important drug which is given to those people who are who had organ transplant. Okay, see, when a new organ has been transplanted, then obviously that organ is identified as foreign. Now, just to see that the body does not react with their own organ and keeps fighting all the time, there is something called as a immunosuppressive agent that is given. Now, this immunosuppressive agent that is very commonly used is a chemical which is called as cyclosporin. Cyclosporin, it's a class of sporins which we have many types. Cyclosporin A is the most commonly used immunosuppressive agent, and this is produced by a fungi which is called as Trichoderma polysporum. Trichoderma is very commonly seen in our soil. It is very commonly seen in our flowing water, and this organism is isolated. Cyclosporin A is uh, produced and kind of, you can say, completely purified, and that is being used as a drug. Okay, so this is one drug which is called as cyclosporin A. Have you heard of one more chemical which is called as statin? Statin is again one particular chemical. This is generally taken for people who are having high levels of blood cholesterol. So if their cholesterol levels are too high, so they are given this group of chemicals which are called as statins, and over a period of time they have noticed that the blood cholesterol level comes down. It lowers the blood chemical level, blood cholesterol level in the blood, obviously. And this statins are group of chemicals, and all this is produced by one of the yeast fungi which is called as Monascus purpureus. Okay, it is produced by an organism which is called as Monascus purpureus. This is a very, very commonly used drug to control cholesterol right now. Now again, here, since we're talking about the good aspects of microorganisms, I would not tell you the bad effects of statins, but nevertheless you should just know that statins for a long period of time have its own bad effects, and also it should not be taken without a uh, proper doctor's prescription. Anyway, so you have this particular uh, statins which stops the production of cholesterol, which stop the synthesis of cholesterol inside the body. So these are just some of the examples where microorganisms are being extensively used, and all this are uh, using large huge fermenters where the media is given, the proper nutritive media is provided, and the conditions are kept so proper that there is growth of either this Saccharomyces or Butylicum or any other organism. Then what we do is growing for some time, and then we are trying to control its uh, what you can say growth by taking them out and then trying to uh, extract them. So this is what we are going to do in the case of use of microorganisms for various purposes. Now with all this, let me just tell you, it is not used only in microorganisms are not used only in industrial purposes or in the commercial purposes. These are very helpful to keep our environment clean, to keep our surroundings clean. In fact, without microorganisms, we would not be able to survive at all, because the whole world would be filled with just dead bodies. So if that is the condition, let us see how these microorganisms are used in sewage treatment. Okay, so let us see how this microorganisms are used in sewage treatment. Now the first thing that we should know is sewage is something which no one wants. It is the waste that is generated, and generally you see that it will have lots of waste like vegetable waste, animal waste, human excreta, and all this. Now the problem with this municipal waste water, which is also called as sewage, is that it has two important components. One is that it has organic matter. Now you might think, what is the big problem with organic matter? Now the second one is with respect to the presence of certain microorganisms, the microbes. Now because there is organic matter and there is also microbes, that is where the problem comes. The problem is that these microorganisms are not all the time good. We spoke about maybe last class how important microorganisms are, but not all the time all the microorganisms are good. Many of them are pathogenic also. Now when we have pathogens and when we have organic matter, these pathogens would feed on this organic matter, start growing, and they would digest the organic matter, of course, but aids the growth of all the pathogenic bacteria, and that would kind of infect us. In fact, it is because of this reason that we should always think, where is the wastewater that we are disposing every day going? Okay, can we dispose it into rivers or streams? We cannot do so because it already has these two components and both of them are bad. And basically what we're trying to do there is, you might be completely polluting the entire river or a lake. So that is not allowed. So what is it that I can do here? Now here, before I treat or before the disposal of the sewage water, we need to treat the sewage water. What do we mean by treating? We have to see that the organic matter comes down and the microorganisms comes down. And this is done in certain units which we call as STP. This is not standard temperature and pressure, but here it is sewage treatment plants. Now in sewage treatment plants, we make them less polluting. Okay. So what do we do here is we try to take out all the types of wastes. We will take out solid waste, we will take out liquid waste, we will take out everything, and then we are trying to carry out a methodical removal of waste and see that the organic matter comes down and the microorganisms comes down. Now how do we do that? That is done by a three-step process: the primary step, the secondary step, and the tertiary step. So let us study about all these treatments uh, one after the other. So let us talk about the first step, which is called as the primary treatment. What do we do in primary treatment? This is the first treatment. So the first treatment always involves taking out the particles. It might be large particles, it might be small particles. We can do, we are removing these particles. And this is done by a process of filtration and sedimentation. Depending on the size of the particles, depending on how much of each type of waste is there, we are going to use filtration or sedimentation. So first what we do is we take a huge tank, and in the huge tank there will be some floating debris, okay, like dead leaves and twigs. So we are going to remove them. Then what we do is, but there would be soil and small pebbles which would settle down. We are going to take them out, and then we take the supernatant. Now this supernatant is ready for the second treatment. So floating one is taken out, the settled one also is taken out. The settled one we call them as the primary sludge. Okay, we call them as primary sludge. So after I do this, taking out the sedimented one and the filtration one, what I get is a effluent which is ready for secondary treatment. So that would be my secondary treatment, and that is the second step in the sewage treatment plant. The secondary treatment is also called as biological treatment. You know why it is called biological treatment? Because here we allow certain microorganisms to grow. Okay, you know what we do here? So from primary treatment we take the primary effluent, and this primary effluent is passed in a huge tank. Now this huge tank will have aeration, so we call it as aerated tanks only. Okay, we call them as the aerated tanks. Now through the aerated tanks we give them enough air, and sometimes we mix it also. Now because of this there is growth of certain microorganisms, and this aerobic microorganisms we call them as flocks. Okay, because they grow in clumps together. Sometimes it's not just the bacteria, you see bacteria with fungi, and all of them they form a mesh like structure. Okay, now when these microorganisms grow, what happens is they eat this organic matter present in the primary effluent. Okay, they eat the organic matter in the primary effluent, and this reduces value which we call as the BOD value. It reduces the BOD value. BOD is the biological oxygen demand. Okay, this is reduced, and because of all this, you see the organic matter comes down to very lower level. Now when the organic matter comes down, automatically you see that the microorganisms also will not get enough oxygen, will not get enough nutrients for its growth, and thus BOD basically is telling us how much of organic matter is present in that water. Okay, if the organic matter is less, then you see BOD is also less. Okay, the greater the BOD of wastewater, then it has more polluting effect. So like this we are trying to take out the organic matter in the first place. Then what we do is we are going to allow this water to sediment itself. Now when it sediments, you see all the bacteria that was grown here, which is forming these flocks, which is growing in these clumps, you know, these filaments, all this will sediment. and this sediment is what we call as activated sludge. Here it was called as primary sludge, here it is called as activated sludge. So activated sludge is again used as a starter culture, again back to the aeration tank, and the remaining one is kind of burnt. Now this water, the effluent that comes here, so what we have grown now, aerobic organisms. Now what we do is we try to grow anaerobic microorganisms. Now when we do this anaerobic growth of microorganisms, you see that so many gases are produced like methane, hydrogen sulfide, carbon dioxide, and so on, and all these gases are used for making biogas. Now this secondary effluent comes out of the secondary treatment after aerobic treatment and anaerobic treatment. Now this can be released into rivers and streams. But however, we do not plan that. What we do is we go for a third step, which is called as a tertiary treatment, where we take extra care to take out the microorganisms also, take out some of the minerals and all that, and we use it for potting the plants. Okay, so that way you see microorganisms might not be useful in primary treatment or in the tertiary treatment, but the whole of the secondary treatment involves growing microorganisms in aerobic condition and anaerobic condition, right? And this is considered to be the best part for the sewage treatment. I cannot come up with any new other novel technology without the use of microorganisms. In fact, you might be knowing that there was something called as Ganga Action Plan, Yamuna Action Plan. All these are used to keep our rivers clean. And all this, just next to Agra, I guess, there is a huge STP sewage treatment plant that is being done, where we are trying to treat the sewage that is discharged into the rivers. So this is a very nice uh, process to learn and to appreciate how beautifully there is evolution, to see that the whole world is in balance. So this is how microorganisms are being used in sewage treatments also. Now the next thing that we need to know is microbes, which are used in the production of biogas. But that part we would be doing later in our next class. So I hope you have understood and you do not have any doubts. Any doubts, make a note. We will always be there to clear them in our live class. See you then.